So good evening all the participants and our today's speaker Dr. Ashwini ji. He is not only a speaker, this terminology is also given by him, and this is third lecture under this banner he is going to deliver today. He is working at Sharda University as assistant professor. Before that he worked at Nimrana University, and he did his PhD from NIT Raipur and postdoc from IIT Delhi. So I welcome Dr. Ashwini ji for generously providing or sharing your knowledge and time for upliftment of this science.
Thank you so much, sir.
And knowledge of bioscience. Now, so on behalf of all the participants, I heartily welcome you, and we all are obliged to you, grateful to you for sharing your knowledge.
Thank you so much, sir.
Anytime, sir. Most of the participants are old, but those who are new, so this is a free webinar series. Anyone can attend. And if you are attending, so you must write your feedback to receive e-certificate. If you are not attending, you are not writing this feedback, then you will not receive any e-certificate. So you can convey to your student, your friend, your colleague also, so that knowledge pool will increase. That is purpose of the Science Dialogue series. So now, Ashwini ji, now
sir,
platform is yours.
Sure, sir. I'm just sharing the slides. So is this visible?
Yes, yes, visible.
So now?
Yes, it's visible, but not in full slide mode. Full slide mode कर दीजिए इसको.
जी sir. Is it visible now?
जो full slide mode होता है ना, complete screen. आप entire screen share किए हैं क्या?
जी sir. Sir, एक मिनट.
Slide show mode में करिए ना.
सर अभी ठीक है सर?
हाँ हाँ पूरा ठीक है अभी.
Thank you, sir. Okay, so very good evening to all the participants who joined this session. So I will start with a very basic question that even I asked to my teachers almost 20 years back, and I get to hear the same question even now when I am on the other side of the table now. When I am teaching my students, I get to hear the same question: what are the scopes in biotechnology? This is not a very straightforward question in the sense that there is no single answer to this. But being a research-driven domain, biotechnology has a lot of scopes, I can say. But yes, it also requires a lot of patience. This session is about how biotechnology has been helping us to fight cancer, and this is just very simple example against just one disease, and there are numerous diseases in the world that we may not know even now. So let's get into the session. I hope you all have heard the word cancer and tumor. So there are always a lot of DNA damage happening inside the cells, and the cell has a very beautiful mechanism to repair the DNA damage. And throughout the day, there are multiple reasons because of which there are DNA damages, and the cell keep on repairing those DNA damages throughout the day. So it is a very active form of repair mechanism going throughout. In case the cell is not able to repair the damage, the cell either undergo apoptosis or senescence. In case there is disturbance in the apoptotic signal, that means the cell is not able to undergo apoptosis, those damaged cells become cancerous. And now, because of multiple mutations in those damaged cells, they gain abnormal cell division capacity, and those abnormal cells become cancerous and create problem for the human body. Now, in case the cancer or tumor is present in the tissue of origin, it is obviously a problem for the tissue and the body. But in case those cells from the tumor side, primary side, detach from those tumor and move to other place of the body, creating a secondary tumor, that movement of tumor cells or cancer cell is known as metastasis. And all the metastatic cases are more problematic than the primary tumor cases. If you can see this picture, the second picture, these are certain hallmarks of cancer. So the first hallmark of cancer is abnormal cell division. That means they stop responding to the breaks of the cell cycle. They are insensitive to the growth suppressors, while they are extremely sensitive to the molecules which promote that cell division. They gain immortality because they don't respond to the apoptotic signal. They may also gain the power of metastasis because there is a lot of physical activity. Uncontrolled division of cell requires a huge amount of physical activity, and for those physical activity, the tumor cells require extra nutrition and blood. And for this purpose, the tumor cells create a lot of extra blood vessels around them, so that they can get more nutrition and blood compared to the normal cells around them. Now, tumor microenvironment is a term that is used to denote all the cells and the non-cellular component surrounding the tumor tissue. This tumor microenvironment is the niche where the tumor cells actually grow. So it's not only the capacity, the inherent capacity of the tumor cells that is responsible for their growth and the problem they create. In fact, they modulate the cellular niche in a way that all the cells and non-cellular component around the tumor cells, they also help the tumor cells to grow. So we have a lot of cancer-associated fibroblast. We have modified ECM, extracellular matrix. We have a lot of immune cells, especially T cells, macrophage, NK cells, dendritic cells, which are there surrounding the tumor cells but are not able to kill the tumor cells, because tumor cells find a lot of different mechanisms to escape the immune system, and that is where the problem starts. Otherwise, if the immune system is capable of killing the tumor cells, it's not a problem for the body. And that's why there are multiple ways through which a tumor cell or a cancer cell can escape the immune system of the body. Okay, coming to the cancer therapies, we all know that chemotherapy, radiotherapy, and surgery are the conventional mode of therapy. Where in chemotherapy we administer a lot of drugs which are cytotoxic. Cytotoxic in the sense their primary mechanism is to kill the rapidly dividing cells. Now we all know that tumor cells or cancer cells are rapidly dividing, so they will definitely be killed by these chemotherapeutic drugs. But otherwise, there are a lot of other rapidly dividing cells in the body which becomes the primary target of these cytotoxic drugs. And if you can recall, the first adverse effect or side effect, that is commonly the word we use, the first adverse effect which is seen in most of the chemotherapeutic cases is loss of hairs, because hair follicles are one of the most rapidly dividing cells in the body. Coming to radiotherapy, we use a lot of ionizing radiations to kill the tumor cells. When radiotherapy started, again similar to the chemotherapy, it used to kill a lot of normal cells around the tumor site. But currently we have a lot of site-directed radiotherapies options available. But still, because the ionizing radiations are being used, there still has a lot of damage to the normal cells around the tumor cells, and therefore radiotherapy is also associated with a lot of adverse effects. Surgery is primarily for solid tumors, and so surgery is required when if a tumor is below a threshold dimension. If the dimension of the tumor is beyond the threshold, it is firstly reduced, the size of the tumor is reduced using chemotherapy and radiotherapy, and then the oncosurgeon go for the surgery. But there are again a lot of challenges associated with the oncosurgery. Then we have targeted therapy, which is very broad term. In fact, we use a lot of small molecule drugs and a lot of antibodies for targeted therapy. Immunotherapy, you may say, is one of the targeted therapy, but there are other components in the immunotherapy which don't come directly under targeted therapy. Then we have a very interesting kind of cancer therapy, which is called where we use oncoviruses. So these are special virus that only infect and lyse the tumor cells, leaving all the healthy cells unharmed. Okay. Coming to some adverse effects of chemotherapy, if you can see the picture on the left side, these are some of the acute and chronic adverse effects. There are many which are not reflected in this picture. If you can see the picture on the right, so it's a cartoon taken from a Twitter post. It's called Chemo Man. So if you can see, there are a lot of chemotherapeutic drugs mentioned here and their primary adverse effects given along. So we can see doxorubicin and carboplatin. Alopecia is one of the most common side effects. Daunorubicin is a cardiotoxic drug. If you can see tamoxifen, it changes the visual activity and corneal opacity. So there are a lot of acute and chronic adverse effects associated with a lot of most of the chemotherapeutic drugs. Okay. If you have heard of this scientist, Dr. Paul Ehrlich, he was working on finding some antibiotics, and there is a term given by Paul Ehrlich which is called magic bullet. So with magic bullet, he actually referred to certain molecules which can only kill the pathogen because he was working on microbial pathogens. So he referred to the molecules or compounds that would specifically kill the microbial pathogen but would hardly harm any normal cell. Now, this concept of magic bullet actually came to cancer therapy with this particular compound. It's a drug which we use, and it is called tamoxifen. The name of the drug is tamoxifen. And if you can recall the targeted therapy from the previous slide, this is the first small molecule drug which is used as targeted therapy for tumors. Now, when we talk of target therapy, there are a lot of small molecule drugs. Tamoxifen, the first example. There are other molecules such as imatinib and erlotinib, sorafenib. These are tyrosine kinase inhibitors. We have a lot of monoclonal antibodies that bind to specific receptors on the cells and exert their action. We have a lot of monoclonal antibodies that release the brakes on the cells, on the immune cells, so that immune cells can normally kill the tumor cells. Okay, I hope you have come across this particular image. This is the basic image of recombinant DNA technology or genetic engineering that we know. So it's very simple. We take the gene of interest for which we want a protein or a peptide. We take a suitable vector, we cut the gene of interest and suitable vector with the same restriction enzyme, and then we add both and we get a recombinant vector. We put the recombinant vector into a suitable host. If it is a peptide, we can use a prokaryotic host. If it is a heavy protein, we must use a eukaryotic host. So we put the recombinant vector into a suitable host and we let the host divide a lot in terms of numbers. And then after a particular threshold, we break open the cells of the host and take out the protein. We purify the protein that we want actually, and then formulate the protein as a medicine. Now this recombinant DNA technology is actually behind all the biotech based medicines we use against cancer. Now the first class is of monoclonal antibody. Monoclonal antibodies are the, I hope most of the participants know about this. These are antibody which are specific to a particular epitope. And now with the advancement in science, we are able to now create monoclonal antibodies where the CDRs bind to two different epitopes. The CDRs present in the same molecule bind to two different epitopes. If you can see this chart, these are the FDA approved monoclonal antibodies. This is just a small chart. There are a lot of other monoclonal antibodies present in the market. If you can see the second last entry, rituximab, it was the first monoclonal antibody that was approved by FDA for cancer treatment. That was in 1997. So the concept of monoclonal antibody, I would say, is not new. Okay. Now, when scientists saw that we can actually target antibodies, monoclonal antibodies to particular receptors, very specific receptors present on the cancer cell, why not attach some cytotoxic drugs along with the monoclonal antibodies so that when the antibody goes inside the cancer cell or tumor cell, the cytotoxic drug also goes along with that. And inside the cell, the drug will get released from the body of the monoclonal antibody, and the cytotoxic drug will show its action by directly killing the cell. Now, this concept of combining a cytotoxic molecule with an antibody, monoclonal antibody, is called antibody drug conjugate. So in this concept, we bind the cytotoxic drug to the Fc portion of the monoclonal antibody, and this formulation is sent or targeted to the cancer. Now, all the cancer are very different from each other because they express many different proteins on the surface from each other. Though there are a lot of common receptors, but there are a lot of very specific proteins also. Now, when the antibody component of this ADC will bind to that particular receptor, and if there is a receptor-mediated endocytosis, the ADC complete conjugate will go inside the cell, and the linker which links the antibody and the cytotoxic drug, it will be broken down by the cellular machinery, and the cytotoxic drug released will now kill the cancer cell. Now, these, if you can see this image, this is just the timeline of ADCs which are approved currently. The first ADC that was approved by FDA was in year 2000, that was Mylotarg. Though it was withdrawn from the market in 2010, but was again approved in 2017 with certain modification. Currently, we have all these ADCs in the market for different cancer conditions. These are some of the ADCs which are currently in the trial. If you can see the image on the left-hand side, so we have currently almost 99 ADCs in phase 1 trial, while 13 are approved by the FDA for different cancer conditions. Now, scientists again thought when we are able to create monoclonal antibodies which are very specific to particular receptors present on the tumor cell, and they are able to exert their action. Now, if the binding is just between the CDR and the epitope of the antigen, why to create recombinantly the complete bulky monoclonal antibody? Why not just recombinantly produce the CDR region? And those fragments of antibodies which are now currently used for a lot of cancer conditions are called antibody fragments. So the first antibody fragment which was developed recombinantly was a single chain fragment which is called scFv. Currently, we have these antibody fragments which are currently in clinical use. We have bispecific antibodies where the CDRs attach to two different epitopes. And one of the very interesting example of bispecific antibody is called bispecific T-cell engager, where one of the CDR will go and bind to a tumor-specific antigen, and the other CDR will go and bind to CD3 of the T cell. Now, this bispecific T cell engagers or BiTEs, which they are commonly called as BiTEs, they bring the T cell close to the tumor cell so that T cell can exert its activity. Now, if you have studied immunology and if you can recall, T cells secrete a lot of cytotoxic cytokines, but in this case, in case of tumors, tumor cells or cancer cells find ways to keep the T cells at bay. But by using these kind of bispecific T cell engager, they just bring the T cells, cytotoxic T cells, close enough to the tumor cells so that the T cells can exert their effect. Now, if you can see the picture, this is the mechanism. You can see TCE, T cell engager in between. With one paratope, with one CDR, it binds to CD3 of the T cell, and with another paratope, it binds to the tumor-associated antigen. Now, when these T cell and tumor cells come close, T cell proliferate, release a lot of cytokines, granzymes, perforin, and this is how they lyse the tumor cells. These are approved bispecific antibodies which are currently in clinical use. The next very interesting therapeutic approach is called checkpoint inhibitors. So one mechanism by which tumor cells actually keep the immune cells at bay is by expressing certain molecules or proteins on their surface, which when recognized by the T cells recognize or anticipate these tumor cells as a normal cells. If you know, normally immune cells don't kill the normal cells in a normal physiological condition. One of the reason is normal cells express certain proteins on their surface, and when T cells bind to these proteins, they come to know that this is a normal cell and they leave the normal cell as it is. These proteins which are expressed by normal cells are also expressed by tumor cells, and when the T cells recognize these proteins, they fail to recognize tumor cells as tumor cells, while they recognize tumor cells as the normal cells. Now, checkpoint inhibitors are the monoclonal antibodies that release the brake. That means if these monoclonal antibody bind to these proteins present on the tumor cell, or the protein present on the T cell that recognize these proteins, binding of these antibody will not allow the binding of these proteins among the tumor cell and T cell, and the T cell can release cytotoxic cytokines and then kill the tumor cells. If you can see in the picture, tumor cell express a protein called PDL1, which is programmed death ligand 1, and the T cell express a protein called programmed death 1, programmed death receptor 1. So when PDL1 is attached to PD1, T cell thinks that the tumor cell is a normal cell and does not kill the tumor cell. But if you can see in the second picture, we are using anti-PD1 antibody or anti-PDL1 antibody. This binding between PDL1 and PD1 is stopped, and T cell recognize tumor cell as the tumor cell. And if you can see the second picture, you can see the lysis of tumor cell happening. Another protein which is expressed is CTLA4. So the earlier two proteins was PDL1 and PD1. The third protein is CTLA4, but the mechanism is same. So currently we have monoclonal antibodies which are against CTLA4, which are anti-CTLA4 antibody. When they bind to CTLA4, they enhance the cytotoxic activity of the inactive T cell and promote the inactive T cell to become cytotoxic T cells. There is a fourth protein which is called LAG3, and the mechanism is the same, and there is currently one monoclonal antibody approved against LAG3, which is called Opduolag. Now, another component of anti-cancer therapy, which I personally feel is a very beautiful gift from biotechnology, is cellular therapy. So, in cellular therapy, we actually modify the cells, immune cells, which are around the tumor cells and which fail to recognize tumor cells as tumor cells. They populate the tumor cells, but they are not able to kill the tumor cells. Now, the first cellular therapy that came to the market is CAR T cell therapy, which is called chimeric antigen receptor T cell therapy. Currently, we are also working on, a lot of scientific groups are working on chimeric antigen receptor NK cell therapy and chimeric antigen receptor macrophage therapy. Okay, CAR T cell therapy is one of the hottest topic in biotechnology today in case of anti-cancer therapies. So if you can recall the previous slide, we discussed about tumor microenvironment. And the tumor microenvironment consists of a lot of T cells which reside there but are not able to kill the cancer cell or tumor cells. Now in CAR T cell therapy, we take out the T cell from the patient. We genetically modify the T cell in a way that these T cells express certain protein on their surface. Now these proteins are not natural. We modify the gene sequence, we prepare a gene sequence, we recombinantly send the gene sequence inside the T cells isolated from the patient. Now these T cells, when express this recombinant gene sequence, it will create a recombinant protein on the cell surface. Now these engineered T cells are proliferated in the lab, and upon reaching a certain threshold in terms of number of T cells, these T cells are infused back to the patient. Now the complete cycle of extracting the T cell, modifying the T cell, and sending the T cell back takes around 30 to 35 days. And when these engineered T cells go back to the body of the patients, because these recombinant proteins which are now expressed, these recombinant proteins are called chimeric antigen receptor. Now, these chimeric antigen receptors are trained to recognize a very specific tumor-associated antigen present over the surface of the tumor cell. Now, these engineered cell, when infused back into the body, they will only recognize and kill the tumor cell, leaving all the normal cells unharmed. So, this picture is not of CAR T cell therapy. This is about tumor infiltrating lymphocytes. So the T cell present around the tumor is actually extracted, is just proliferated in the lab and infused back to the patient body. In this case, in case of TIL therapy, we don't engineer the T cell. We are just enhancing the proliferation capacity of the T cell in the lab and then infusing back the T cell back to the patient. These are some of the, so this is the first TIL therapy approved by the FDA. These are some of the TIL therapies currently in different phases of clinical trials against different types of cancer. And this was about the CAR T cell therapy that we just discussed. Now, currently we are into the fifth generation of CAR, in the sense that the first generation of CAR, it expressed a kind of protein on the surface of the T cell, which was not very accurately enhancing the capacity of the T cell. But with the fifth generation of modified gene of the CAR, currently all the CAR T cell therapies which are there in for the clinical use are extremely specific to the tumor for which they have been developed. But currently all the CAR T cell therapies which are there in the market are only for blood cancers or different types of leukemia. But yes, scientists are working very hard to develop CAR T cell therapies for solid tumors also. One reason for currently why we don't have any CAR T cell therapy for solid tumor is again tumor heterogeneity. Tumor heterogeneity is a term which is used for the condition where the cells of the solid tumor are very they are very heterogeneous in nature. That means if there are 10 different types of cells in a solid tumor, all the 10 different types of cell will express different types of proteins. Now, if we choose only one protein to target by modifying or by generating a kind of CAR T cell, other 9 cell types will remain unharmed or will not be killed by that particular CAR T cell. And that's why tumor heterogeneity is one of the major reasons why initially CAR T cell therapy was only developed for blood cancer and not for solid tumors. But again, with the advancement of biotechnology, currently we are developing a lot of CAR T cell therapy for solid tumors as well. These are the CAR T cell therapies currently in use in clinic. But unfortunately, all these 6 CAR T cell therapies are not present in India because of financial reasons. A single infusion of these 6 CAR T cell therapy, any of these 6, will roughly cost around 4 crore of Indian rupees, which is practically not feasible for most of the people in India. Even if the government support through a lot of different government schemes, it won't be possible for government also to spend 4 to 8 crores of rupees for a single patient. Currently, most of the patients, obviously not in India, who have received these 6 CAR T cell therapies have shown very good results with single infusion. But in case some patient require double infusion, it will cost around 7 to 8 crores of rupees, which is practically almost impossible for anyone living in India. Okay. Now, these are some of the CAR T cell therapies which are currently in different phases of clinical trials, and these are for different types of tumors. You can see. The second picture is for CAR T cell therapies in clinical trial for different solid tumors. In fact, okay, this was a very recent news where a CAR T cell therapy was used in trial in certain children who were suffering from one deadly type of brain cancer. And if you can see this picture, this protein GD2 against which the CAR is developed, GD2 is actually the protein for which the news was there. So if there is an expression of GD2 in a brain cancer, it is considered as one of the deadliest type of brain cancer. Okay. Now, as we just discussed that the initial 6 CAR T cell therapies that are there in the market, obviously not in Indian market, they cost around 4 crore of rupees for single infusion, which was not possible for most of the Indians to afford. Now, this slide I'm going to share is one of the most favorite stories of mine, which is a very beautiful example why indigenous research is very important. If you can see the picture of this scientist, he's Dr. Rahul Purwar. He's currently associate professor in Department of Bioengineering at IIT Bombay. Now, he's the person who developed the first indigenous CAR T cell therapy in India right from the scratch. The gene sequence that he developed for the CAR protein is completely different from the gene sequence that have been used for the earlier 6 approved CAR T cell therapies. Not only this, with that improved gene sequence of the the CAR T cell therapy that he developed, he and his team developed, uh, also shows very minimum adverse effects that were very prominent with the earlier 6 CAR T cell therapies. Some of the most common adverse effects were neurotoxicity and cytokine release syndrome. And with NextCAR19, the name of the product that was developed by Dr. Rahul Purwar and team, this product NextCAR19 show very minimum neurotoxicity and cytokine release syndrome. So this was the team. If you can see the name of the scientist headed by Dr. Rahul Purwar, Dr. Alka Dwivedi and Dr. Atharva Karulkar were the PhD students who were involved in development of this product right from the scratch. And this NextCAR19 is the first CAR T cell therapy approved in India, and it just cost around 30 lakhs of rupees for single infusion. So you can compare from 4 crore to 30 lakhs how indigenous technology and indigenous research can bring down the cost of the products. Currently, we have just discussed this product, NextCAR19, which is now Rahul Purwar is also the founder of a biotech startup called ImmunoAct. It is a Mumbai-based startup. So ImmunoAct, this company now manufactures NextCAR19. Currently, we also have a company called Immunil, which is funded mostly by Dr. Kiran Mazumdar-Shaw of Biocon. And Immunil has Professor Siddharth Mukherjee in its board as a co-founder. And the second CAR T cell therapy right from the scratch developed in India is Quatemi. And very recently, it has also approved by CDSO for use in Indian patients. Now, we have just discussed a few minutes back about the tumor heterogeneity. So in a solid tumor, we have different types of cells which express different types of proteins. And therefore, because of this heterogeneity, it is difficult to develop cellular therapies against this. But still, the beauty of science is that science always goes ahead. And with the advancement of science, we have a lot of CAR T, CAR NK, and CAR macrophage therapy against solid tumors. So these are some of the targets and CAR products used for CAR macrophage therapy. These are so this is a comparative chart between CAR T, CAR NK, and CAR macrophage. In case of CAR T, the T cell is always isolated from the patient itself. But in case we are trying to develop CAR NK or CAR macrophage therapy, the NK cell and macrophage can be allogenic also, and can also be developed from induced pluripotent stem cells. These are some of the CAR NK cell therapies which are currently under clinical trial for different types of cancers or tumors. So these are just the tables. These are the CAR-M therapy, CAR macrophage therapy, which are currently under clinical trials, various phases of clinical trial for different types of tumors. Okay, the last point or the last kind of last therapy that I would like to discuss is oncolytic virus. These are the virus which specifically target the cancer cell, proliferate inside the cancer cell, tumor cells, and lyse them. So this is the basic mechanism of direct killing of tumor cells by the oncolytic virus. A second mechanism by which oncolytic virus kill the cancer cell is by affecting the tumor microenvironment. So in this case, in the second case, we genetically modify the oncolytic virus with protein that would go and inhibit a kind of cytokine which is called TGF beta. TGF beta is one of the most prominent cytokines present in the tumor microenvironment, which is responsible for the resistance of the tumor microenvironment. Now, when these genetically modified oncolytic virus are targeted to a specific tumor, they will when they will go inside the tumor cells, they will express a protein that will inhibit TGF beta. Now, when TGF beta will be inhibited, the T cells, the cytotoxic T cells and NK cells, macrophages, dendritic cells present in the tumor microenvironment, they will become active and can then directly lyse the tumor cells. There was a very interesting recent news where a scientist whose name is Dr. Beta Halisi, she's a virologist who was working on oncolytic virus. Unfortunately, she was diagnosed with a stage 3 breast cancer, and rather than going for conventional therapies, what she did, she injected herself with the oncolytic virus she was working on. And fortunately, for last 4 years, she is completely free of the tumor. Though this is the motivation lies not behind what she did, but what she is actually doing with her research. So these are some of the oncolytic virus which are approved currently for different types of tumors. If you can see, most of them are for melanoma, and one product is for head and neck cancer. Okay, so this is a very beautiful statement. I very often say in my class also, and very strongly believe that today's science is tomorrow's medicines. Whatever research, the fundamental research scientists are doing, will someday see the light of clinical translation. So thank you everyone for the patient hearing, and I'm open for the question.